All right, hello students. Welcome to our week 3 lab. So today we're going to be going over cell structure and function. All right, so cell theory originated back in the 1600s when an English scientist actually first described the appearance of plant cells under a microscope. So when he looked in the microscope, the structures he saw reminded him of the cells of a honeycomb, and so he named them cells. If you think about a honeycomb, they have all these regularly spaced holes. So essentially, this is why we call them cells. So this picture here is a series of images of a cell undergoing mitosis, which we will cover in a few weeks. The red is stained, the red color shows the chromatin, which is the chromosomes, the DNA, basically the genetic material of the cell. And then the green is tagged to show the cytoskeletal filaments, which is what pulls each set of chromosomes into a new daughter cell. So I just love this picture. It's just very beautiful, and it'll give you an introduction of what we're going to look for in a few weeks. So essentially, cell theory states that all organisms are composed of one or more cells, and that the cell is the basic unit of structure and organization in organisms. And also importantly is that cells can only come from other cells. So because we are biologists, we love to So because we're biologists, we love to classify things. So because we do that, we have two different kinds of cells, a prokaryotic cell versus a eukaryotic cell. So the word prokaryote actually means before nucleus. And this is basically a term that explains that these organisms arose in the evolutionary time scale before the evolution of a nucleus. So these cells don't have a central nuclei, and they don't have organelles. So basically there's a nucleoid, which is just a general area of the cell where the DNA is. Most prokaryotes are in the bacteria and archaea kingdoms. So they do have some morphological items that are pretty common across these kingdoms, but not every single prokaryotic cell has it. So they do have a cell wall, and they do have external structures like fimbriae or flagella. In the picture you see the single flagellum coming out of our little green bacteria there. And they also have pilus or pili, which are also external structures which help bacteria move and sense things in their external environment. And there is a capsule which surrounds these cells, and this capsule consists of the glycocalyx. So the other type of cell we have is the eukaryotic cell. So eukaryote means true nucleus. So in eukaryotic cells, we see the evolution of compartmentalization in the cell. So this increases the efficiency of the cell because we can then segregate different biological functions into these organelles, which helps to increase efficiency. And in a few weeks, we're going to learn about the peroxisome, which helps to rid the cell of hazardous hydrogen peroxide. So it has this function compartmentalized in an organelle, which protects the rest of the cell from some highly toxic substances. If you just think about it, think about your body. Stomach acid is in your stomach, and your stomach has evolved lining to deal with that. But if we had stomach acid going through your entire body, obviously that would not be very pleasant, and it would not be it would not help you survive. So this is what happened basically with the eukaryotic cell. All right, so parts of the eukaryotic animal cell. So we have organelles, which are all these little structures. So this should match the illustration that is in your lab notebook. So this will help you go through that figure and complete it. And then if you also look at table 4.1, there are several structures that are described that are very important for the cell. So this diagram contains more than you will actually need to know, but it's still important to look at. So just some highlighted organelles that I think you should focus on. We have the nucleus. So this is what contains all of the DNA, the genetic material of the cell. We also have the endoplasmic reticulum. So there's two types of endoplasmic reticulum. We see the rough ER and the smooth ER. And what differentiates these two structures is that the rough ER is basically studded with ribosomes. So these ribosomes are embedded into the membrane of the rough ER. If you basically just think about just a very complex series of membranes, and these ribosomes are in the membranes, and it really just looks rough under the microscope. So then there's also the smooth ER, which lacks ribosomes, so it doesn't appear rough, it appears smooth, and these ribosomes are not embedded in this portion of the ER. So ribosomes are some of the most important structures in the cell because these are the particles that carry out protein synthesis. If we didn't have ribosomes to make proteins, we could not exist. They make just about everything that we need in the cell. So they have ribosomes that are embedded in the rough ER, which complete protein synthesis there. And then we also have ribosomes that are free-floating throughout the cell, where they complete protein synthesis for things that are meant to stay within the cytoplasm. The ribosomes in the endoplasmic reticulum generally synthesize proteins that are going to be exported to either the membrane of the cell or externally to the cell. All right, so next we have the mitochondria, or singular mitochondrion. So it's true, the cliche is true, the mighty mitochondria is the powerhouse of the cell. So these are the organelles that carry out cellular respiration, which produces ATP molecules. ATP is the currency of energy for a cell. And so I won't go into the mitochondria and how it functions, but it is actually a very cool organism. I might post something about the mitochondria in case you want to delve into this, especially the electron transport chain, which is very fun. But if you are interested in how exactly the mitochondria does this, I'll post something for you to look at. But if you just want to leave it as the mitochondria generates energy, that's fine. All right, so moving on, we also have the Golgi apparatus. So this looks like basically a stack of floppy pancakes or a bunch of flattened sacks that are next to each other. So the Golgi apparatus is basically the sort of the mail sorting center of the cell. It processes, packages, and secretes modified cell products. So as proteins are synthesized in the rough endoplasmic reticulum, they eventually move into the Golgi apparatus where they get modified and transported and packaged for transport to wherever they need to go to the cell, whether it's the cell membrane or if it's secreted by the cell to the external environment. All right, we also have the cytoplasm. The cytoplasm is a semi-fluid matrix that's outside of the nucleus, and it contains organelles. So it's not basically an inert fluid. There's a lot of research going on right now about the cytoplasm and how the cytoplasm actually can separate different regions of itself. So it's a very interesting topic of research. All right, we also have vesicles, which are these small bubbles next to the Golgi. So these are the vesicles that will come off the Golgi. They're membrane-bounded sac that stores and transports substances. So if you can see there, you can see that the Golgi is sort of secreting this vesicle to the cell membrane as it moves out. All right, another organelle that's important is the lysosome. This is a personal favorite of mine because the topic of autophagy that I study uses the lysosome to digest macro molecules and cell parts. So this lysosome is, you could kind of think of it as the stomach of the cell, in that it's going to break down all of these substances in the cell. So sometimes there are just old organelles that don't work anymore, or just proteins that aren't needed by the cell anymore. And so the cell basically chops these things up in the lysosome. It's a pretty toxic environment. The pH will decrease to digest it, similar to the way our stomach acid digests things. And then these materials can be recycled by the cell, because the cell, is very lazy. It wants to exist using the least amount of energy possible. All right. So we also have the centrosome and the centrioles. So these are organizing centers that help to organize chromosomes during mitosis, and we'll talk a little bit more about these structures in a few weeks. And then we also have the cytoskeleton. So like I said, the cytoplasm is not just some inert fluid. There are these filaments that work its way through the cytoplasm, including microtubules, intermediate filaments, and actin filaments. And these fibers all work to move things around, and it's actually a very interesting structure. We won't have to go into this in too much detail. You don't need to know all these different filaments, but just know that the cytoskeleton exists. It helps to maintain the cell shape and assists movement of the cell parts through the cell. And then also we have the plasma membrane. So the plasma membrane is very important. There really would not be life if we didn't have the plasma membrane. It's the outer surface that regulates entrance and exit of molecules into and out of the cell. And it actually has these proteins embedded in it. It's not just, a film or a balloon. This is actually a very highly complex structure. And I posted something about the cytoskeleton, a little activity for you guys, for you folks to work on. I thought it was a great video. Really just explains how complex the plasma membrane is. And so even though we cannot do our dialysis bag experiment today, with moving the sucrose and the starch, or the sugar and the starch into and out of the membrane, you can still think about the different molecules and how they might transport across a membrane. All right, so parts of a eukaryotic plant cell. So plants are also eukaryotes. They have a lot of structure. They have organelles, many of the same organelles that we just went over in the animal cell. So I'm not going to go through this, all of these, but I am going to highlight the two very specific organelles that are important for plants. Actually, three. So first we have the cell wall. So the cell wall is a highly structured portion of the cell that surrounds the outside of the cell membrane. So the cell membrane, the plasma membrane is still there, but we also have the cell wall. And this is really important because you think about plants, they need to stand up straight, especially trees. Think about how tall that is. If they were just mushy membranes, they would not grow to be 30 to 40 feet. So the cell walls add structure to the plants. We also have the chloroplasts, so these big green organelles. They look kind of like vitamins. They look like green vitamins. These are the chloroplasts. And chloroplasts are where photosynthesis occurs in the cell. So this is how plants are able to harness the energy from the sun in the form of photons, and they can perform a complex series of chemical reactions which turns that energy into sugar. So this is not the energy producing portion of the cell, it is the food producing portion of the cell. So the chloroplast can then feed that sugar substance into the mitochondria, same as animal cells, and the mitochondria then produce ATP to energize the cell. So it's important. You need both the chloroplast and the mitochondria in plant cells. It's not one or the other. And then the third important part of the eukaryotic plant cell is that central vacuole. So you see this giant structure in the middle. which is a large fluid-filled sac. It stores metabolites and helps to maintain turgor pressure. So the cell wall is important for structure, but the central vacuole also really, really helps to keep the plant solid. So, if you have any house plants or if you garden at all like me, you can kind of see this in action when you see a plant that needs water. What happens to a plant when it needs water? It sort of sags. It's very droopy. It doesn't stand up straight. So the central vacuole, once you water the plant, the cells are able to take up water, fill up their central vacuole, and they're able to stand up straight. So, take a look around your house. If you don't have a plant, you should get some plants because they're awesome. And see how water can affect that plant structure. All right, so diffusion. So diffusion is a very important process in biology. But it's also a very inefficient process in biology. And so the cell basically works to overcome this. So the basic principle of diffusion is that there's a general movement of molecules from a higher concentration to a lower concentration until equilibrium is achieved and the molecules are distributed equally. So if you look at the picture that I'm using, you see dye molecules. If you drop, say food coloring into water, at first it'll go in as sort of a drop, and then eventually it spreads out throughout the water until it reaches equilibrium, and then you will have an evenly colored solution of water. But at first you could see those ribbons. Actually, I might demonstrate this for you downstairs in a bit. All right, so then what are we talking about in diffusion? We're talking about solute, which is a substance being dissolved. So in this example, it would be the dye molecules. This can also be salt or sugar. If you think about dissolving salt, not salt in your tea, sugar in your tea. Don't put salt in your tea. That's disgusting. It takes some time for it to be dissolved, for you not to see those sugar crystals anymore. And this is just how the sugar molecules break down until they're equally distributed in the water or the tea. So that would be the solvent, the substance in which the solute is dissolved. All right, so diffusion in the plasma membrane. And like I mentioned, you should go through that video that I assigned because it will really go into much more detail than we have here. So homeostasis, which is the ability of an organism to regulate its physiology and survive, depends upon the plasma membrane. If we just had substances going into and out of our cells, we would not be able to survive. Just think about the things you come in contact with and how if they were just able to enter your cells, no problem, you'd be in some trouble. So some molecules can diffuse across the membrane and some cannot. So as you can see, there's a lot of substance in the extracellular fluid, and some things can pass through the plasma membrane, but this largely depends on its size or its charge. So osmosis is basically a very specific term for the diffusion of water. And so we usually think of water as a solvent, but water can also act as a solute. It just depends on what which molecules movement you're monitoring. So like other solutes, water will also follow its concentration gradient and move from high concentration to low concentration. So if you're thinking of water as the solute and say the sucrose molecule as the solvent, water is going to move across that membrane into the compartment that has the high level of sucrose molecules because it wants the concentration of water molecules on either side of this membrane to be equal. So water will move to the right into the compartment with the sucrose molecule. So eventually this compartment will swell because you will need more water molecules in there in order for it to be equally distributed. So tonicity refers to the relative concentration of solute and solvent across the plasma membrane, so inside of the cell compared to the external environment of the cell. So there are three different terms that we use when we're talking about cellular tonicity. We talk about the isotonic solution. So iso means same. So an isotonic solution means that there is the same concentration of solute and therefore of water as the cell. And when cells are placed in the solution, there is no net movement of water. So if you've ever been to the hospital and you need fluids in the form of an IV, they will give you an isotonic solution. So if they put pure water into your veins, you'd have an issue with your cells, because our cells don't exist in pure water. We have, we have salt in there, lots of ions. So you'll get a saline solution. So this saline solution is isotonic to the amount of saline in your blood. A hypertonic solution is when there's a higher solute concentration, than the cell. So when cells are placed in a hypertonic solution, say you put a cell into a very salty solution of water, water is going to move out of the cell and into the solution to try and equalize the distribution of water molecules across that membrane. So when this happens, you'll see a phenomenon known as crenation, where cells will shrivel. So if you look at the hypertonic image in my slide, you see that the cells sort of just look mushy. They don't look like nice plump blood cells, because the water has left the cell, so they're not as solid. So in the opposite solution, we have the hypotonic solution, in which there is lower solute concentration and therefore higher water concentration than inside of the cell. So this would be the situation if you just put pure water into your IV. When cells are placed into a hypotonic solution, water is going to move from the solution and into the cell. And because the plasma membrane, it has elasticity, but there is a breaking point, these cells can sometimes burst. So this is called hemolysis, which the cells will swell and burst. And if you look at the hypotonic solution, you could see the cells are looking very puffy, and some of them are actually bursting, which is not good. We need red blood cells. So we've seen what tonicity does to cells that just have a plasma membrane, but what happens in cells that have a cell wall, like a plant? Does tonicity affect these cells? And the answer is yes, it does. So turgor pressure. So I had spoken a little bit about well-watered plants and how they can stand up straight and they're not droopy. In a hypotonic solution, the large central vacuole will gain water and exerts pressure called turgor pressure. So if you look at the hypotonic solution, you can see that vacuole is super full and it's pushing against the cell wall. So if you think about it, the cell wall is not going to move much because the pressure from the water is going to be pretty high. So you could think of this would be a very, very firm, rigid cell. And then in the opposite, where there's a hypertonic solution and water wants to leave the cell to equalize the concentration of water across the membrane, you're going to see the central vacuole shrink, and the cell membrane is going to pull away from the cell wall. So the cell wall is a rigid structure. It's not going to move. It's not going to shrink like we see in animal cells, but it will shrink, and you'll see this. You could actually see the plasmalized cell where the plasma membrane pulls away from the cell wall. And so in the lab, we usually do this. We take Eladia, which is a freshwater plant, and we place it into salt water, and you can actually see that the cells pull away. So another way of doing this is I could use potatoes. So I'm going to slice some potatoes and show you all how that works as well. All right, so pH in cells. So this is another reason why compartmentalization of the cell increases efficiency. So the pH of a solution indicates its hydrogen ion concentration. So basically that means whether it's more acidic or less acidic. So the pH scale ranges from 0 to 14. 7 is neutral. It's neither too acidic nor basic. It's just water. Water is a pH 7. Most of our enzymes in our body function around a pH 7. So the lower the pH, it's more acidic, and the higher the pH, it's more basic or alkaline. So like I mentioned before, our stomach acid is very acidic. It has a very low pH, around a 1. And then there's also a higher pH. And so later on in our digestive system, we actually have to increase the pH to be able to dissolve lipid molecules. And so the stomach is just very interesting how it can adjust its pH dramatically. So a higher pH would be base or alkaline. So this would be your bleach would be the upper end of the spectrum. A bleach or sodium hydroxide have about a pH of a 13 or a 14. So buffers. So pH can affect the physiology drastically. We're going to talk about enzymes in a couple weeks, and we'll talk about how pH affects the activity of enzymes. But for most of the enzymes in the human body, we need a pH of 7.4. Otherwise, these enzymes will not work, and your body will stop functioning. So we are actually very happy between a pH of 7.35 and 7.45. And any movement out of this range, whether it's towards the acidic level or the alkaline level. So we have acidosis, which is when your blood pH is low, and when we have alkalosis, when your blood pH is high. And if you go too far, basically the body will cease to function, and you will die in either way. So this can happen with several different diseases, acidosis or alkalosis. So it can happen. So buffers are a way that we are able to control the excess hydrogen or hydroxide, which is the opposite of hydrogen towards alkaline, ions. So a buffer will essentially soak up these excess ions, either hydrogen or hydroxide, and they're designed specifically so that they can alternate between either of these to keep the pH in the appropriate range. So antacids, this might be something that you could do a little bit at home or just think about. So stomach cells, like I've said, they secrete hydrochloric acid. So if you've ever had heartburn, it is very painful because that hydrochloric acid is seeping up into your esophagus, and there's no protection in your esophagus for that acid, whereas we have mucus and lining in the stomach, so you start to feel that pain. Antacids will neutralize the acid in the stomach that causes heartburn. So, it counteracts the acid. It basically moves it to a more neutral pH, and then it stops hurting. All right, so we have, these are the lab exercises that we would have been completing in our class. I'm going to do the potato strip experiment and show you how salt concentration affects the rigidity of the plant cells. You would have also visualized the difference between prokaryotic and eukaryotic cells. We had prepared slides, so you won't really be able to do that, but I have posted some electron micrographs of cells so you can actually see just how complex the different structures are. They actually look really cool. So then another thing we would have done is to prepare and analyze the wet mount of the Elodea leaf cell to visualize the cell wall as discussed. And then we would have observed diffusion through a semi-solid, which was an agar plate, a liquid, and air. The air is actually my least favorite thing to do in lab because we used an air freshener spray and then measured the time of how long it takes for people to smell it. So it was rough on my scent-induced migraines. So we also had the plasma membrane, which was the dialysis tubing experiment. So you can take a look at the plasma membrane video and activity and just think about how things would move across the plasma membrane. And that will be it. So hopefully I will have seen you at office hours today. I'm having office hours every Friday from 1 to 2 now on Zoom. So just click the Zoom link in my contact info. And I will hopefully get some demonstrations going for you now. Hi students. All right, I'm doing some kitchen science. So here's my setup. So I've got salt, a potato, some water. I'm also going to do a little bit of the food coloring experiment just to show how diffusion happens through a liquid. All right, so here's our setup. So I've measured out in these two containers 100 milliliters of filtered water. I just use my Brita filter water. It's not deionized, but you know, it's better than tap. And then here I just have regular tap water, which I'll be doing the food coloring experiment. So, got some blue food coloring. Oop, which I just got all over my hand. Now I have the cap off. So with blue food coloring, so if you can see, so I put 2 drops. As you can see, the food coloring stays together, but eventually will diffuse throughout this entire glass. So we'll let that sit there for a little bit and see how long it takes. All right, I've sliced up my potato. So, you know, it's pretty firm. This one's pretty firm as well. I think I'll take another slice. All right, so I have 2 slices of potato now that are about the same size. You know, they're not exact, but I'm trying to control as best I can. So now I just need to make the salt water. All right, here is my kitchen scale because like everybody else in the world, I also was trying to make sourdough starter during lockdown, but I failed every time. Okay, so to make a 10% salt solution, we're gonna add 10 grams that's 11 grams, close enough. All right, so this one on the side will be our salt water. Just gonna swirl that up so it dissolves. All right, I've labeled both of these with a pen, and now I'm going to drop my potato in. Yeah, so here's a little bit of a squish. Okay, you know, it's pretty firm, a little bit pliable. Whoop. That one's in the water. Again, you know, pretty pliable. That one goes in the salt water. And we'll be back in an hour. All right, it's been about an hour, we're back. So as you can see here in the water, it's mostly completely diluted. You know, very different from what we saw when we began with. You can actually see on the bottom that there's a little bit of the dye that collected in the bottom. I think that's just because the dye is heavier than the water and so it sank. But, you know, I think if we left this here for a while, that would disappear and you would see a completely uniform concentration of the dye. All right, let's check out our potato. Haha, it's stuck. All right, so here's our potato that was in the water. Pretty tough, actually. You know, it's really not that flexible. All right, we got the salt. I'm really bad at it. All right, here's the salty potato. Yeah, it's like really floppy. You know, you can see that I can really mush this up. So there you have it. The salt from the water in the solution, in the hypertonic solution, drew out the water from the potato. See, this is the water one, so I cannot, you know, whoop, see, it snaps. And this more sort of just mushes. So you can see that the cells have lost a lot of water. And just as a reminder, this is the one that was left out that I cut. All right, so there you have it. I did some science at home. And that should help you complete at least some of the tonicity questions for the lab. All right, thank you everybody for watching my kitchen science. I will post the questions that you need to complete in the lab manual in the text of the video. All right, have a nice weekend. Bye.